are the mainly the two types of the responses: the primary response and the secondary response. The primary response, in this, there are the millions of the B cells and each synthesize one particular antibodies. These immunoglobulin molecules are inserted into the cell membrane of the B cells, and these are the known as the antigen receptors, and the antigen binding site is displayed on the cell surface. And these are here the antigen binding receptors in the B cells, and the B cell displaying their antigen receptors. The antigen possesses specific epitopes, and each epitope is recognized by the complementary antigen receptors. Here we can see that there is how this is recognized. The one epitope is recognized by this complementary part of the epitope present on the antigens, and these are the paratopes that is present on the immunoglobulins. And the B cell displaying their antigen receptor here. And when an antigen enters for the first time, its epitope is recognized by some of the immunoglobulin molecules, and these Ig molecules bind to the antigens. And after this binding stimulates the B cells to divide and differentiate. And the B cell differentiated into a clones of the plasma cells, and these plasma cells mm, release out the further more of these types of the antibodies. First, here we can see the antigen select the complementary receptor on the surface of a B cells. After that, the antigen bind to the antigen receptors, and these B cell divide and differentiated into the plasma cell. Uh, After the several signaling pathways, and the clone of the plasma cells are secreted in, in the form of antibodies, neutralizing the effect of these antigens. And the plasma cells not only synthesize but also secrete the antibodies. And these antibodies secretion upon first exposure to the antigen is called the primary response, and this response is a weaker form, and it is of the short-lived one. Whereas the secondary response, this is the response that uh, take place after the encounter of the first response. Here, the first encounter with antigens, some of the B cells are converted into the memory cells. So, after if the same antigen enters against the memory B cells differentiated into plasma cells, this is called the secondary response. Secondary response is quick, strong, and long lasting. Here with the graph, we can see here the antibodies level are increasing. In the primary response, we can see that the antibody level is not increasing at that much. After the second exposure of the same infections, there is a secondary response which is very hyper. This hyper reaction is taking place due to the memory cells, and these memory cells are having the antibodies. These antibodies uh, giving the more exposure to all the other cells, and so they are drastically increased in the antibodies level can be seen in the secondary response.